Hantavirus is a disease. It's an infection. And like many other infections, due to the COVID-19 epidemic, it's flown under the radar. But I'm Nicholas Ryan, and I'm going to tell you some information and why you should care about hantavirus. I'll start off with the basics. The formal name of hantavirus is orthohantavirus, and some common names it's known by are simply hantavirus, hantavirus pulmonary syndrome, also known as hantavirus cardiopulmonary syndrome, and a subset known as hantavirus hemorrhagic fever with renal syndrome. The infectious agent of hantavirus is based on its attachment of virions to cellular receptors and then subsequently endocytosis. But this is just the beginning. Nucleocapsids are introduced to the cytoplasm by a pH-dependent fusion of the virion with the endosomal membrane. Then transcription of the viral genes occurs in association with L proteins and the 3 nucleocapsids. In addition, transcriptase and replicase functions. The viral L protein is also thought to have endonuclease activity. The viral N and L mRNAs are thought to undergo translation at the free ribosome, whereas the M mRNA is translated in the endoplasmic reticulum. The L protein produces nascent genomes by replication, and the hantavirions are believed to form by association of the nucleocapsids with glycoproteins embedded in the membranes of the Golgi apparatus, which are followed by this budding of the Golgi cisternae, which then allows the nascent virions to be transported through secretion to the plasma membrane and then released by exocytosis. The taxonomy of the hantavirus begins at the realm. So the realm is Riboviria, kingdom Orthonaviridae, phylum Nergarnavircota, and Eleovircota, Bunyelviralis, and Hantaviridae, finally getting to the genus of Orthohantavirus. Many other hantaviruses fall all under this. There's dozens of different hantavirus out there. Viri, I guess. But let's start looking at the characteristics of the infectious agent. Like the name suggests, the hantavirus is in fact a virus, but it consists of 3 single-stranded RNA segments, which replicate exclusively in the host cell's cytoplasm, where the genes are encoded in the genome complementary sense. So, complementary, yeah. The sRNA encodes the nucleocapsids protein, and the mRNA encodes the polyprotein, which is co-translationally cleaved to yield to the envelopes of the glycoproteins. And hantavirus, like I said, replicate exclusively in the host cell cytoplasm, but the entry into the host is thought to occur by attachment of virions to the cellular receptors. Eventually, this produces nascent virions, which are transported via exocytosis. Some notable strains of hantavirus are Cinnobre, Prospect Hill, Pumala, Seoul, and Hanten. As opposed to what I listed here, I'm actually going to start at Hanten, because Hanten's kind of interesting. It was the first strain isolated of the hantavirus by the Hanten River in Korea. Its reservoir is striped field mice, and it is typically found in Asia. And many soldiers in Korea were actually infected and afflicted with the hantavirus before we even knew what the hantavirus was. Cinnobre is found in deer mice in North America, whereas Prospect Hill, its reservoir is the Meadowvale, and results in no human disease, and it's found within the US. Pumala's reservoir is the Bankful, found in Northern Europe, and the Seoul hantavirus reservoir is domestic rats and is found worldwide. So hantavirus does not just afflict a singular part of the world, it is a global issue. The viral antigens found in hantavirus are found mostly with endothelium cells of capillaries, and almost all components of the infection infect endothelial cells, and that's how we are most commonly able to determine whether or not something is infected with hantavirus. And the methods that hantavirus goes through to impair its host are quite complex, because it seems as if the hantavirus is going to kill its host before it's capable of replicating. And it's quite interesting, but at the end, the features of the hantavirus suggest that the mechanism of inflammatory cell recruitment in the lungs specifically of hantavirus pulmonary syndrome patients results from specific attraction and adherence of selected population of inflammatory cells to an activated pulmonary microvascular endothelium. And this is based on the capillary permeability of the lungs. Transmission of the hantavirus is very basic and simple. Contact with rodents. These are the zoologic carriers of the hantavirus. Their droppings or urine are a source of transmission, and you can even inhale particles in an area of concentrated feces or urine. And not to mention, but the direct contact with a human host also infected with hantavirus of any form can lead to transmission. Prevention, like transmission, is also simple. Rodent control is the primary source of prevention for hantavirus. And an increased rodent population increases the chances of contact with an infected rodent host. And so avoiding disturbing the dust when cleaning out previously infected areas after rodent control and removal has occurred is key in preventing possible infection because of those aerosolized particles. The at-risk population of hantavirus is basically anybody that comes into contact with rodents, and that increases the risks of contracting the hantavirus. Rodent infestations in and around the home remain the primary risk for hantavirus exposure. Individuals who stick out and may be more at risk are campers, hikers, utility workers, construction workers, and pest control workers, because those individuals are spending more times in fields, out where rodents live, and in old abandoned buildings where they may be nesting. Symptoms of hantavirus basically cover the board for every sign of illness ever. They trigger a humoral response in the human body, which results in fever and chills, blurred vision, low blood pressure, intense back pain and stomach pain, rash, shortness of breath, nausea and vomiting, coughing, dizziness, tiredness, etc. And in order to diagnose hantavirus, in regards to HPS, it really involves knowledge of contact with a potentially viable source of infection. So an individual spends a lot of time in their barn, they've noticed rats, they tell their doctor, hey, we've got rats in our barn. Then the doctor can move forward from there and diagnose them with hantavirus. For hemorrhagic fever and renal syndrome, laboratory tests can be done, and they utilize the clinical history, again, the knowledge of rodent exposure, to determine whether or not they could potentially test positive for hantavirus. And then they perform serology, so they take blood and they look for a positive for the hantavirus infection and the antigen present through their immunohistochemical staining and microscope examination. But RNA sequencing can also be performed in place of this, and that is the method in which they go about diagnosing hantavirus today. Now, treatment for hantavirus, there's really no specific cure or treatment for hantavirus infections. And those that are diagnosed early on are typically placed into intensive care, intubated, and given oxygen therapy to help them get through a series of severe respiratory distress. But that's just for the hantavirus pulmonary syndrome. For those afflicted with the hemorrhagic fever and renal syndrome, the management of the patient's fluid is crucial, and their electrolyte levels, and they maintain the correct oxygen blood pressure levels through oxygen therapy. And in order to treat appropriate secondary infections, they typically use antibiotics, but in early onset cases, they typically use ribavirin, which is one of the only medications used to treat HFRS. Current research efforts are done focused on the behavioral changes of people rather than rodents, and how to adjust society in order to mitigate chances of infection, such as McConnell 2020 suggests that countries with awareness, like Chile and Panama, decrease the infection rates as opposed to have not. As opposed to in the US, Arizona has they have had significant outbreaks of the hantavirus since 2017, I believe. They have had 78 confirmed cases of the hantavirus. And that's compared to 2 entire countries. The future outlook of research and hantavirus is that previous research hasn't yielded a cure or a vaccine to hantavirus, but that just means the future is one of opportunity. Nobody has created an effective vaccine, and it has been present in human populations for probably hundreds of years. So why can't one of us cure hantavirus?